Hello students, welcome to my genetics video for instructional segment 4 in our Living Earth NGSS curriculum. I hope you have enjoyed it and learned a lot, and I encourage you to have your study guide handy and to go through practice problems as you're doing this. Let's jump in. Like all my videos, this is not for profit. It's solely meant to help students and anybody that comes across it on the internet. All right, Tazen, let's do it. The standard that we're working on is applying concepts of statistics and probability to explain the variation and distribution of expressed traits in a population. This ties into DNA, into evolution, and many of our other units, and I hope you can make a little concept map and see those come into play. Please check out all the work you've done in Google Classroom. We did a gizmo, we've done stations, we've done various homework assignments, we've had packets, we've had simulations. All of those are good items to review. And if you are thinking about taking the AP class, I'd recommend you try those AP challenge questions on genetics and go through some of the problems that I have here. The phenomenon that we started this unit with was labor congenital amarosis, and this young lady named Molly. And Molly, unfortunately, inherited two recessive alleles that were going to make her blind. And so, these recessive alleles are marked with a little red spot here. Both her mother and father were okay because they have the enough of the dominant protein, this blue one, that their eyesight is okay. But Molly didn't inherit a dominant allele. She inherited two recessive alleles, and those recessive alleles are denoted in red here. The actual gene that's affected is called RPE65, I believe, and it's a protein that works in the back of your eye cells. Luckily for this story, we had a good ending in that Molly received some gene therapy and a modified adenovirus that carried some copies of the correct protein, this blue one, into her cells, and her eye cells are able to express that gene therapy product there. And so there's a lot of work on this happening right now from Jennifer Doudna and I believe it's Emmanuel Charpentier, who won the Nobel Prize for CRISPR on the ability to maybe fix some of these diseases and try and rewrite DNA. Very exciting times, something that you could study in college. So, once again, big picture, she was going to be blind, but due to gene therapy, she is okay and can see. And that we're really trying to see this interplay between genetics and DNA and proteins, which we studied in our last unit. A gene is a segment of DNA that codes for a protein, and you may have different versions of genes, and they may have dominant or recessive characteristics, or they may not. They may be codominant or incompletely dominant, etc. So always come back to the phenomena and see why Molly had these eyesight problems, and let's dive on in. Here is a picture of, it's called a karyotype. So a simulated picture of human chromosomes here. And this is just to help you review a homologous pair of chromosomes. This would be homologous pair number 1. You would get one of the chromosomes from your mom and one from dad, and one from mom, one from dad. Each of them have the same genes on them, but they may have different alleles or forms of the gene. You have about 30,000 genes, so you have lots and lots and lots of genes on each chromosome. The last pair of chromosomes, number 23, denotes gender or biological sex. And you could be XY, which would come out as phenotypic male, or XX, which is phenotypic female. But in class, when we get to it in meiosis, we will study that there is a range of differences in sexual development, and that there's differences in what comes out as the phenotype there. Okay, so let's kind of get into it with the study guide. I'm going to do a little bit reviewing Mendel before I hit section 1, that vocabulary. So let's jump in. Can you describe Mendel's experiments right here? Pause the video, explain it to your brother or sister, your parents, your friend, your dog, whoever is around. All right, hopefully you got something along the lines of Mendel was a monk who did these meticulous pea plant experiments, and we really call him the father of genetics for this. So he had this pure breeding tall stock, right? And so they always, always, always came out tall in those seeds. And they needed a pure breeding short stock. And these always, always, always came out short. So this would be called the P generation. He then bred the tall and the short. He meticulously did this, transferring pollen between them, and he got all tall. This is the F1 generation. So it looks to him like the tall wins all the time. So now when you breed the tall by the tall, you end up getting 3/4 tall and 1/4 short. But the short disappeared in this generation and comes back out here. What is going on? My goodness. So this would be the P generation up here. Let me see if I can highlight it or write it. P. This is the F1. And this is the F2 generation. If genetics was a blend, right, then the plants would have come out medium and stayed medium forever. If tall was dominant, and he thought they might come out all tall all the time, but they come back out here. So let's see what's going on genetically. Here's that blending model prediction. Here's Mendel's actual results. Oh, one thing to note here, his actual numbers are 787 tall, 277 short. So it's not quite 3/4 and 1/4, but it's pretty close, right? 3/4 would be 750 and 1/4 would be 250. So it is a probability. Okay. So here he is crossing them, making sure to be very, very, very careful to not have cross-contamination, etc. So here we go. From that little review, can you nail some of this vocab? And you might need to pause the video. You might need to look at your homeworks. You might need to really think about it. All right, so go ahead and do that. Okay, let's go through them here one by one. We'll check them off as we do them. A gene. A gene is a segment of DNA that codes for a protein. This is the height gene, whether the plant is tall or short. This is the texture of the pea that it makes, whether it is a wrinkled pea or a smooth pea. That is the gene. An allele is a form of a gene. This plant has in its homologous pair of chromosomes 2 tall alleles. It is homozygous tall, but it has 1 wrinkled and 1 smooth allele. So it is heterozygous for smooth. Smooth is dominant over wrinkled. Okay, so that went over dominant and recessive. A dominant allele is expressed even in the presence of a recessive allele. A recessive allele is only expressed for the plant if it receives 2 copies of the recessive. So the only way we would get a wrinkled plant is if instead of smooth, if this was also wrinkled. Then we would have 2 recessive alleles. One thing that's important to note, especially as you get to higher level biology, this wrinkled and smooth, they are actually both read by RNA polymerase and made into mRNA and made into proteins in a cell. It's just that the smooth protein is what you see. So the wrinkled protein is either beat out in a pathway, or the smooth adds up to this phenotype, but they'd actually be making both proteins. Just like here, it makes both tall proteins. Okay, heterozygous means 1 dominant, 1 recessive, or different alleles. Homozygous, the prefix homo means the same, so here this would be 2 dominant alleles. A gamete is just going to be a sex cell, and the sex cell from this person, this plant, that is, thank you, that is homozygous tall and heterozygous smooth, once again, was big T, big S, little s. A sex cell or a gamete is only going to have 1 allele of each gene. So the gene here had 2 dominant alleles, but it's only going to put one in a sex cell. And let's say it puts the big T and the little s, right? You don't get to choose. Or it could be a big T and a big S. Well, this is the way it works. When you make sex cells, they only have one allele in it for each gene. And then when they combine together with your partner sex cells, then the baby's going to have two alleles for each gene. And that's called the haploid-diploid life cycle. And so this enables that the correct amount of genes are passed on to each generation, or the correct number of alleles, which would be two for each gene. Okay, so we've done gene, allele, dominant, recessive, heterozygous, homozygous, gamete. The P generation, F1 generation, and F2 generation were back in the previous slide, right? P being that pure breeding group, F1 being the first generation or the cross between the two P stocks, and then the F2 being the F1 cross by an F1. From those three generations, we can figure out if a trait is dominant or recessive or what's going on. Phenotype stands for the physical trait. It's what you see. So we might see whether it's tall or smooth. Genotype stands for what is the actual gene. So the big T, or the big S, little s. Those would be the genotypes. A Punnett square divided by original Punnett is what we use to show the probabilities of inheriting certain traits. And it is the same, or it's the famous box with 4 here. And so if we're crossing these, we would put the dominant allele. These on the outside represent gametes or sex cells, big S, little s. I kind of drew it backwards. All right, homologous chromosomes. Here's a homologous chromosome, one from mom and one from dad. So you get one allele from mom and one from dad. An allele is a form of a gene. And the principle of segregation says that only one allele goes into a sex cell. It's random which one it is. So for this plant in its sex cell, it can make wrinkled or smooth, but only one goes. All right, let's continue on. I hope you found that helpful. Here's just another picture of Mendel's work. And so here were the true breeding plants for flower color, for purple and white, or violet and white. When they were bred together, they all came out purple. But then when he bred the purples by the purples in this F1 generation, you got purple and white. So that white came back out. If you think about this as DNA, right, a gene is a segment of DNA that's being passed down, then it should make sense, hopefully for you, that these had one purple gene and one white gene or segment of DNA. And in here, when they're crossed together, this one got two white genes or two white segments of DNA. Here's the actual traits that he used on his flowers to figure all this out. And here is an example of it from Khan Academy, actually, and the OpenStax textbook, which is excellent. So here is that P generation, so yellow and green. Due to the principle of segregation, in their sex cells, they only have one allele. And because they're homozygous, it's always going to be a big Y for this one, for the yellow plant. And for the green, it's always going to be a little y, because that's all it can give. It is homozygous. Now the F1 generation comes out yellow. And we watched a TED Ed on this, Mendel's Peas on Earth. That was pretty fun. So now when we breed the F1 by the F1, so in other words, we're breeding big Y, little y by big Y. It's important to draw them so that you can understand them. Little y. Here we're going to get 3/4 yellow and 1/4 green. The principle of segregation says that the gametes, half of them will get this capital Y, the dominant version, and half of them will get the lowercase y, or the recessive version. And these are the probabilities. So the genotypic ratio would be 1 homozygous dominant, capital Y, 2 heterozygous, big Y, little y, and 1 homozygous recessive, little y, little y. The phenotypic ratio would be 3/4 yellow is what we see, and 1/4 green. All right, oops, let me clear my screen here. Thank you. This is called a test cross, and we'll go over this more in class. But here, let's pretend that we have a yellow seed, but we misplaced it. And we don't know if this seed is from the P generation or the F1. We don't know if it's pure breeding yellow, that it's always going to come out yellow, or if it's heterozygous, big Y, little y. If we want to figure it out, we can do what's called a test cross by breeding it with a homozygous recessive. It always has to be homozygous recessive. If it comes out that all the baby plants are yellow, then we know that this one had to be big Y, or dominant allele, because all the resulting baby plants came out yellow. If they come out half and half, then we know that this one had to be dominant and a recessive, or heterozygous, a big Y and a little y. And that's because then you would get half and half. So this is where it becomes fun, right? You got to use probabilities. You have to use reasoning. You have to use critical thinking skills to figure out what's going on. All right, we're ready now to do a little bit more with the study guide. So here comes some sample problems for you to stop and pause the video and check them as you wreck them. Hopefully you got this. This is called a monohybrid Punnett square, where we're going to be analyzing one trait at a time. Remember that, so for this one right here, let's look at this box right here, students. It's going to be bring that dominant B over, bring the recessive B here. We normally write the dominant and then the recessive, the big letter and then the small letter. Just it's easier for us to read, but it doesn't have to be that way. Your body doesn't care. Your body could read the little b, big B first. Here comes how the Punnett square works with an animation by the Amoeba sisters. All right, and so here you can see that recessive. Here you can see the genotypic ratios, and then you can see the phenotypic ratios. It would be, let's see, well, this is a different one, so this would be half and half. All right. Here was the original one, so this would be 3/4 hairy and 1/4 non-hairy. All right, what about two traits that runs, the whole dihybrid? So this is where we're going to really move beyond a middle school understanding to more of a high school understanding of genetics. You are 30,000 traits at once, right? You're 30,000 different Punnett squares going on. So let's start, though, with something a little more simple. Here is the yellow and round and green and wrinkled seed. And this is what almost all biology classes use because it is what Mendel did, and it's a very classic, famous experiment. When he crossed them, all the F1 generation was yellow and round. So that tells you that those are the dominant alleles, right? Due to the principle of segregation, this plant can pass on a big Y and a big R in its gametes. Notice how it's only one allele for each gene in the gamete. This one can pass on little y, little r. Therefore, all the F1 plants are going to be big Y, big R, little r. Now we're going to cross those, but we got to figure out the possible gametes first for big Y, big R, little r. Can you do that real quick? Well, the way you do it is what's called FOIL. Is the easiest way for us to remember. Gametes go on the top and sides of the Punnett square. Sex cells can only have one copy of each allele. So our math trick was FOIL. So let's go ahead and do it. It's going to be first. First is big Y, big R right here. Outer, big Y, little r. Excuse, first, outer, inner, last. Yes, outer, I'm sorry. Oh, they just did it a little different. But it still gives you all the characteristics. So we're going to go big Y, big R, big Y, little r, inner, little y, big R, last, little y, little r. So that's a good example of me needing to have checked my picture before making this. And also that it really doesn't matter what order you put these in on the outside of the Punnett square, as long as you have all the possible combinations. So for our class, I taught you and went through it with you a couple of times. the foil method. So here, let's see it in action. There, that was pretty good. First, outer, inner, last is the way I remember for doing these 2 trait crosses. So here's all the possible gametic combinations. Notice here for this homozygous recessive for both traits, the only sex cells it can make are the recessive version. Why don't you go ahead and practice? Try this out. Draw it on your paper, all right? Draw one big box and then split it with 3 lines and cross with 3 lines. That should give you 16 boxes. All right, here's what you hopefully got. So we did first, outer, inner, last, and here are the gametes, and you're going to get a 9-3 to 3-1 ratio for these dogs here. What if we don't get Mendelian ratios? Like oftentimes you see people who have red hair have freckles. Well, that means that the genes were linked, and we'll get into that later on in the slideshow. So it has to say, if you're going to use these math rules, it has to say that the genes assort independently. I will not try and trick you with that in 9th grade biology. That's more for AV. Okay. Here is the same thing with the dogs. All right. And so let's see the probabilities here. So what is the probability of getting capital B, capital C, little c for the puppy? Well, what we can do here is we can use the product rule. If it says and, so we want to know the probability of being capital B, little b, or heterozygous and heterozygous. Hetero and hetero. And means multiply. So here we can do the Punnett squares for each trait. So take the B's and put them here on this one monohybrid Punnett square. Take the C's and put them here on one monohybrid Punnett square. Then find what you want. And so here we wanted a probability of big B, big C, little c. That's 1 out of 2 here. Or you could say 2 out of 4. Here would be 2 out of 4. 2 out of 4 times 2 out of 4. You do numerator times numerator, denominator times denominator, would be 4 out of 16. And if you reduce that, it becomes 1/4. That's a lot quicker, students, than doing the 16 boxes, which is why we use the product rule a whole bunch. But I have to assume independent assortment here. So this is a new slide that I added in this year. Like, let's check this out. Here's 4 traits at once. So if we were to go from 2 traits to 3 traits, that would go from from 16 boxes to 64. And then you would have to do it times 4 to get to the next level. So it would be a lot. It'd be a lot of boxes to fill in for the Punnett square, but you could do it, right? And so here I'm asking, what is the probability that the baby will have all dominant traits? So let's use the product rule. So that means dominant A, dominant B, dominant C, and dominant D. All right, we're going to do our A's here. And so I'm crossing this heterozygous A times this heterozygous A. And so this is my result. Here's my B's. I'm crossing heterozygous B, homozygous recessive B. Here's my C's from up here, crossing big C by big C, little c. So homozygous dominant by heterozygous. Woo, they all come out dominant. Then I'm going to cross my heterozygous D by homozygous recessive, and we're going to get this. So let's go ahead and do the math, right? It's going to be 3 out of 4, 2 out of 4, 4 out of 4, 2 out of 4. And if you do that and simplify it, it comes out to 3 out of 16. That's the chance that you're going to have a baby that has all dominant traits. It might not be as high as you thought it was because they all have to hit for that to happen. All right, on to non-Mendelian genetics. Here's my crazy kitty cat in the fridge just to spice things up. Can you describe multiple alleles or blood types is what we did in class. So here you could be A blood, you could be B blood, you could be AB blood, you could be O blood. So there's more than one allele or more than 2 alleles for blood type, right? And so earlier we're doing the plant is either yellow or green. It's either smooth or wrinkled. Here we're taking it that there's more possibilities. All right, we did a whole pogo on this and a couple of warm-up problems, so hopefully you got it. Can you describe antigens and antibodies? This is, here's a picture of it, right? Let's pretend that you are type A blood. So that means you make a type A antigen. An antigen is a marker on the cell that elicits an immune response. You don't make immune responses to your own antigens. This is your self antigen A. You would make an immune response if you got B blood. So you would make an antibody for B blood. So if you're type A, you cannot receive type B blood because you make antibodies for it. Antibodies look like little Y-shaped things. And so the antibody that you would make as a type A person might look like this Y that can grab onto the B spike. Now let's pretend, like Mr. Gibney, I'm B positive. Let's pretend that I don't need to pretend. I do have type B blood. So what would happen if they gave me type A? My own antigen, my self antigen is B, so I am not allergic to that. That's all good. But if they gave me A, I make little antibodies that can grab onto the little circles here. So they might look like little half moon, half crescents, right, on the Y. So that's an oversimplification, but that's how it works. So if I get A blood put in me, even when I'm type B, I will have a bad reaction. My body will take those antibodies, will clump up the A cells, and I will have clots, and I could die. If I'm AB and I get A or B blood, I'm okay because I have both of those antigens. They are both expressed. One is not dominant over the other. So this is an example of multiple alleles, and it's actually an example of co-dominance as well. Both of them can be expressed. Type O doesn't have an A or B antigen. So it is the universal donor. Anybody can receive type O blood because you won't have a reaction to it. Now, it could have a positive or negative marker that's called the rhesus factor. So the true universal donation is O negative. Anybody can receive O negative. If you are O negative, though, you can only get O negative blood. Anything else will cause your body to clump up. All right. We said that it's determined by codominant alleles. Here they are. So you could be type A, type B, AB, O. If you're A or B, you can either be homozygous or heterozygous for that blood type, respectively. So why don't you go ahead and try and draw out all the different types of blood you could be right now. Here's our problem sets that we will do in class that we haven't done yet. Here we go. Hopefully you got something like this. So you could be homozygous or you could be heterozygous. Here, homozygous B, heterozygous B, AB, or O. All right, go ahead and try a problem. Mom's type O, dad's AB. What can the kids be? Woo! Hopefully you got something along the lines of this. The kids could be either A or B. They cannot be AB, and they cannot be O. All right, mom is type B heterozygous, and maybe you know that because you know that her mother, your grandmother, would be O, or granddad. Dad is type AB. What could the kids be? Here we go. Heterozygous B, homozygous, or codominant AB. So the children could be AB, A, BB, or B. So that would be, both of these would be B. All right, how about type B heterozygous, type A heterozygous? What can the kids be? They can be anything, right? They can come out as A, B, B, A, or O. So this one's a really good one to understand. Look at it. It gives you the gametes. It shows you the Punnett square. You should pause the video and work your way through this problem. All right, what are some other exceptions to Mendel's rules? Can you describe incomplete dominance? So incomplete dominance is, I told you the joke, if you turn in something incomplete, nobody wins. Here, neither the red nor the white protein wins. You instead come out pink. This happens in snapdragon flowers. You might say, well, why is this important to learn, Mr. Gibney? Here I got a red and a white, and it comes out pink, 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 pink. Well, if you cross the pink by the pink, if it was the concept of blending, then all the results would be pink. But instead, you get red, pink, pink, white. And so the red and the white pop back out. And once again, shows that genetics is not blending, it's many different Punnett squares put together. You might say that, but Mr. Gibney, that is not true. Look at me. I am a blend of my parents. I, too, am a blend of my parents. My mother was Japanese. My dad's Irish and English. So I kind of look in between. I get it. And that's because we are polygenic. We have many genes contributing to a trait such as skin color, hair texture, eye color, and more going on. So if you're polygenic, and if I have, let's say, 13 different Punnett squares contributing to height, and that it's the summation of those Punnett squares that lead to my height, that is why you look like a blend. But in reality, we're not blending the DNA together. You're having many, many, many Punnett squares going on in your body. So here, this is just another real good example to disprove the concept of genetics blending. So here's that chromosome. Here's that segment of DNA that's going to code for red. Here's that segment of DNA that's going to code for white. And they pop back out when you get that homozygous version of them. Red, red, white, white. But if they have both, then it's incompletely dominant. It looks like a blend, and it is pink. Okay, can you describe codominance? So we saw a bit of that with blood types, but you do it here. So this is called a Rowan cattle. So it can either be red or white, or both of them can be expressed, and it's red and white. So in both incomplete dominance and in codominance students, the heterozygous form is the one that exhibits the incomplete or codominant trait. Can you describe sex-linked genes? Here is the kind of classic case of hemophilia and a carrying mother and an unaffected father. So the Y chromosome in our class right here doesn't really carry very many genes. It carries the SRY gene that helps the code for being a male, but it doesn't carry the other genes that the X chromosome does. So men are more likely to inherit and express sex-linked diseases. A female has two X's, so she, if she is a carrier of a disease, won't express it, right? The dominant, or the X with the dominant version, will be expressed and she would be okay. So here it is for hemophilia. She has hemophilia is when your blood can't clot, so you can't stop bleeding. And so this mother is a carrier. All of her daughters will be unaffected. Her daughters will get from dad an X with the dominant allele, and from mom an X with either the dominant allele or the recessive. So one half of the daughters will be XX with two dominant alleles, and one half of the daughters will be XX with one recessive allele, one dominant allele. So they will be carriers of the disease. So if you were to say out of all children that this couple could have, it would be 1/4 female with no carrying of the disease, 1/4 female carrying the disease. And then half of the kids could be boys. And so from dad, they get a Y. All sons get their Y from their father. This son gets this X from mom, the X with the capital H or the dominant H. And this son, unfortunately, gets this X from mom, the X with the recessive H. You don't get to choose which X you pass on unless you do like in vitro fertilization or something. You don't get to choose. Otherwise, we'd always choose the best for our kids. So here, these would be the sons that have hemophilia. So this is the same thing that's at play with muscular dystrophy. All right, so this was kind of figured out by Thomas Hunt Morgan and Alfred Sturdivant with fruit flies. So try this one. Here is a white-eyed male and a red-eyed female. Red is dominant to white. Can you complete the cross? What would be all the possible outcomes? If we got something like this, so here, all the females that are produced will have red eyes, right? Even though they inherit the white-eyed allele from the male, they're all going to be red-eyed females. Here, they're also going to be all red-eyed males because they're going to get a red allele from the female. So this is an interesting Punnett square. You took, a test tube full of white-eyed males and a test tube full of red-eyed females, and it all came out red-eyed. Well, now let's breed this F1 generation. So these are going to be heterozygous red-eyed females by a red-eyed male. So heterozygous red-eyed females by a red-eyed male. What do we get? Bam. Now we get to something a little more interesting. We're going to get, this one's going to be red-eyed female, XX. I told you to do the X's first. This one's going to be a red-eyed male, X with the red allele, and then Y. If you're wondering why they use a W, that W stands for wild type. It's what you see in the wild. So in the wild, normally the flies have red eyes. Here, XX with one red allele. So this female will come out red-eyed. So all the females have red eyes. But here, the male comes out X with the recessive, with the mutation, with the white-eyed, and then the Y. This is going to be a white-eyed male. So this cross produces an interesting 3/4 red eye, 1/4 white eye, and is showing that the white-eyes are only males. And so that's how they figured out that it's a sex-linked trait. The females never came out with white eye. Now, never is a strong word, right? There can be a mutation in the creation of sperm or egg that could lead to a recessive allele. It's just rare. All right, that was hemophilia. Here is a calico cat. And so this the one I put in the slideshow at the beginning. Here's my cat Lion, named after Dr. Mary Lion. And what Dr. Mary Lion showed was X-bar inactivation. So females inactivate one of their X's in each cell early on in embryonic development. You actually don't want a double dosage of the X proteins. Because coat color for these cats is on the X chromosome, all these ones, this X got inactivated, the black X, and here the orange X was kept active. All the daughter cells of this one will be orange, and all the daughter cells of this one, consequently, will be black. And so here are all the daughter cells that were black for our cat Lion, and here's all the daughter cells that were orange for our cat Lion. And then the white part is a different gene that kind of overrides coat color for both of these. So that's kind of interesting for calico cats. So why can a calico cat not be male? Right? Male cats would either be orange or black. They'd be XOY or XBY. There's exceptions, right? And that could be what's called Klinefelter syndrome. The cat could be XXY. And if that instance, you could have a male calico cat. All right. We're almost at the end. If you're stuck with me, great job by you. These are pedigrees. I think these are super fun. You got to apply reasoning. You have to use critical thinking. I want you to not be too worried on your assessment. If you give me good reasoning, even if you get it wrong, I'm going to give you a lot of the points, right? I just want to see your thinking. So a pedigree shows the passage of a trait in a family. And here would be generation number 1. We label it Roman numeral 1. And then we go across 1, 2, 3, 4. So now if I want to talk about this person right here, I'd say Roman numeral I-3. Here in this generation, I'd say if I wanted to talk about this person right here, I'd say 2-2. Here I'd say 3-1. The boxes are boys and the circles are girls. And I just remember boys for boxes and circles for ladies. Okay. If it is shaded, it means they have the trait. If it is half shaded, it means that they are a carrier. What we do first is we try and figure out, is the trait dominant or recessive? Then we say, if it's recessive, is it autosomal, so on chromosomes 1 through 22, or is it sex-linked? And there can be other instances such as mitochondrial or Y genetics heredity. So let's look at this one. What do you think? Is it dominant or recessive? Go ahead and take a guess. All right, if you got recessive, you are correct. The reason we know it's recessive is because it skips a generation. It skips 2-1 and 2-2. So let's do the counter. If it was dominant, and if this was capital A, little a, that would mean that this one was little a, little a. It would mean that all these children are little a, little a. We know for one, even though one marries into the family, because they don't have the trait, that's impossible. It doesn't work. All right, so it's using reasoning. It has to be recessive. Here is the little a, little a. Here's the capital A. Let's see. Well, hold on. Because this could be, this child is heterozygous, so capital A, little a. So this is capital A question mark. We don't know if they're capital A, or capital A, little a. This person is heterozygous, capital A, little a, and that's how they get this child who has the trait and these children who don't. So because it skips a generation, it's recessive. All right. If we want to see if it's sex-linked, we should look at dads and daughters and moms and sons. So here is a daughter, and here's a dad. If it was sex-linked, then this dad would need to have the trait. So say it's hemophilia, because he would be X, little h, Y, and he would pass on that X to his daughter, and the mother could pass on a recessive X as well, and then she would have the trait. So mom would have to look like this: X, X, big H, little h. And that's how this individual could come out as X, X, with the two recessive traits, little h, little h. So since that doesn't happen here, it cannot be sex-linked. We can rule that out. Now, what's tricky is you can rule out sex-linked, but you can never fully rule out autosomal recessive. You can just say that there is more evidence for a sex-linked trait. All right, let's go on. Here's some from worksheet we did. This is actually from that HHMI activity on pedigrees. And so you would use claim evidence reasoning to try and figure out what's going on here. Sometimes you won't have enough information and you won't be able to say whether it's dominant or recessive. For instance, for number 6, we can't look at that and figure out whether it's dominant or recessive. We don't have enough information. For number 2, we know it's recessive. For 1 as well. For 3, 4, it has to be recessive trait because it skips a generation. So here is an X-linked recessive. So let's see if it fits the pattern. Does it skip a generation? Yes, it skips a generation. So therefore, we know it's recessive. It doesn't have to skip a generation to be recessive. That's just a clue, a giveaway. Let's look at moms and sons and dads and daughters. So here's a dad and daughter. Dad has it. Daughter has it. Let's look for any moms and sons. Oh, we don't have any on here. Okay? So this could be X-linked recessive. Here is mitochondrial. Mitochondrial goes from mom to all of her children, and it's only passed on by moms because only moms give their mitochondria to the baby. There's actually some evidence that it might not be completely true anymore, but we're going to go with it for here as scientists are figuring it out. So here's the egg, and the egg has mitochondria in it. Right, here comes the sperm. The sperm does not give mitochondria. It just drops off chromosomes. So slightly more than half of you is from your mom. So that would be mitochondrial inheritance. All right, see if you can guess for this one. What mode of inheritance is this? This is hard. This is the first one that's dominant, okay? It is dominant. Dominant traits cannot skip generations, so it doesn't skip any. And the way I can tell that this is an autosomal dominant trait is if I look right here, if it was recessive for 2-5 and 2-6, if it was little a, little a, little a, little a here, then all their children would have to have the trait. But because not all their children have it, then it is most likely a dominant trait, capital A, little a, capital A, little a. And here, this child got the little a, and doesn't have it, whereas these children do. There is always the possibility of a mutation happening, but not very likely. And for these instances, we'll rule out mutations here. Okay, here it is with Thomas Jefferson and his sexual assault on Sally Hemings. And so his slave. And then you can see that Sally's children, Eston, he had children, they had children. And so eventually we're able to get Eston Hemings' descendants and look at their Y chromosome. And we could look at Thomas Jefferson's descendants, Y chromosomes. But Thomas Jefferson didn't have any male children that survived to have their own kids. But we know that because his dad's brother or his uncle had children, that we could look at their Ys, because the Y does not undergo a lot of recombination. The Y is passed on intact. It doesn't do the same type of crossing over that other chromosomes do. And we found that it does match up. President Thomas Jefferson did assault his slave Sally Hemings and rape her and have children by her. So that is there, and Monticello affirms this. Some bonus items for you to research are listed here. Genetics is just awesome. There's so much stuff. We could do the rest of the year on genetics. Unfortunately, we just don't have time. We got to move on. The Learn Genetics Utah website is just powerful. I hope you got to check it out and have some fun with it. Enjoy studying this. I think it's fascinating items. We're going to go over more in class. Take care, students. Have a good one. Peace out.